Welcome everyone to today's webinar on integrating classical biological control for spotted-wing Drosophila. The webinar will last about an hour and then we'll have 30 minutes for questions. So today I am excited to introduce 4 members of a national research team who've been investigating different strategies for the organic management of spotted-wing Drosophila as part of a NIFA OREI funded project. James Brown from UC Berkeley, Ben Johnson from the University of Maine, Cesar Rodriguez-Saona from Rutgers University, and Philip Fanning of the University of Maine, who I'm going to hand the presentation over to now, and he's going to give an introduction to the projects.
Excellent. Thank you very much, Alice. Okay, folks, thank you so much for joining us today. I'm going to just give a quick introduction of the project and then we'll get into some of our content. So this is part of an EFA funder project called On-Farm Integration of Organic Management of Spotted-wing Drosophila in Fruiting Crops. Our goal is to reduce the economic impact of Spotted-wing Drosophila on organic food production. And we've been doing this a number of ways. It's been through the development and the evaluation of decision aid tools and thresholds, a subject we're not really going to touch on today. Also integrating the recently approved APHIS biocontrol agent for Spotted-wing Drosophila, Ganaspis camorum. This might have been previously known to you as Ganaspis brasiliensis, but there was a recent name changed. And we're looking at integrating this new parasitoid into existing organic crop management practices. And then finally, we're going to do outreach and education and develop a training program for, we'll say, the content that we develop. Like Alice said, project website is eorganic.info/spotted-wing Drosophila, and that's where you can see some of the other content that we've been generating under this project. So we developed 3 research objectives for this project. The first was to develop and expand monitoring and an economic-based decision aids for organic agriculture. The main one we're going to focus on is to promote beneficial insects in organic fields. And specifically in that objective, that's the one we're updating you most on today. And that's specifically on optimizing methods for release and quantifying the establishment of Ganaspis camorum, determining the impact and sustainable management on natural enemies and pollinators, and then determine the compatibility with organic compounds and nematodes with Ganaspis camorum. So this is a large multi-state project, and we got quite a large team focused on this research. As you can see here, we have researchers from the University of Maine, University of Minnesota, UC Davis, Oregon State University, University of Florida, USDA IRS in Corvallis in Oregon, Michigan State University, UC Berkeley, University of Georgia, Rutgers University, Cornell University, Washington State University, and then of course eOrganic, which is hosting this webinar in Oregon State University. Now, importantly, all of the PIs listed on the previous website have quite a large team that they've been working on. So there's many graduate students, postdocs, technicians, and undergraduate students, and we'll talk a little bit about our undergraduate students later on, that are all involved in making this research happen, and we thank them as we present this research today. It's also important to note that this project is building on the efforts of 2 previous projects, which were also funded by USDA and IFA, and specifically the Organic Research and Extension Initiative Program. Both of these were led by Ash Sial in University of Georgia. And then our current project is also a USDA and IFA funded project. Okay, so obviously if you're here with this webinar today, you probably already know the focus of this webinar is on Spotted-wing Drosophila. This is just a devastating little invasive vinegar fly. It's native to Southeast Asia, and it was introduced into the United States. It was first discovered in California in 2008, and it quickly spread across the country, and at the same time became a global cosmopolitan pest. We call it Spotted-wing Drosophila because the males have those spots in their wings, And importantly, those females have that serrated ovipositor. That means they can oviposit into marketable fruit, unlike other vinegar flies that we have here in the United States. Now, this vinegar fly has really large consequences in terms of its economic impacts. This is quite a small insect, but it has led to huge losses in our fruit industry, specifically here in the United States. This is the results on this slide are from a survey that we ran back in 2014. It showed just how much damage we were getting associated with this insect. So blueberries, it was estimated we were having a 13% crop loss back then. Blackberries, 27% crop loss. Raspberries, which is really a preferred host there, we were estimating about a 41% crop loss. And cherries was only at a 9% crop loss. But giving the value of these crops, these were large losses in terms of the economic costs. Now, importantly, we want to highlight we're also rerunning this experiment at the moment, or this survey at the moment, to see where we are in terms of our losses associated with this insect. So this survey was ran in 2014. We are currently updating the results of that survey, and I'm going to pop a link in the chat here in a minute, which you will be able to click on and take the survey to see or share with us what the losses might be in your production if you are a grower of some of these crops. I will put that in right after I pass it over here. So here's the webinar outline. We are going to first talk about an overview and progress of SWD biological controls, assess do releases make a difference. We're going to cover OMRI approved pesticides and their compatibility with SWD parasitoids, non-target effects of insecticides with and without a phagostimulant on Spotted-wing Drosophila parasitoids. And then finally, we're going to end before our questions with a bit on determining the compatibility of nematodes with Spotted-wing Drosophila parasitoids. Okay, so I'll pass it over to our next presenter, which is Dr. James Brown from UC Berkeley, who's going to present our first section.
Thanks, Phil. Like Phil just said, my name is James Brown. I currently work as a postdoc at UC Berkeley in Dr. Kent Dana's lab. Before coming here, I finished my PhD in University of Florida with Dr. Oscar Liebert. So I've actually had the opportunity to work on 2 of the OREI teams that are part of this program. And I'll be talking about biocontrol and some of the progress that we've made so far. And there we go. So first, a little bit of context about biological control. When we think of biological control, generally, we think of insects predators, like think green lacewings or ants, and insect parasitoids, think of the topic of our conversation today, Ganaspus. But in addition to those biocontrol insects, there are also other biocontrol agents that can be used to manage pests. There are biocontrol bacteria, fungi, and nematodes. And in fact, the OREI group is also working on some of these other biocontrol agents to determine their compatibility with Ganaspus cumorum and to determine how to effectively deploy these agents in service of managing Drosophila suzuki, spotted wing. Over the course of this project, a lot of really great work has come out of learning about biocontrol for spotted wing. On the screen, you see 3 publications from collaborators on the OREI project. These publications do a really great job of summarizing the progress that we've made thus far on all the biocontrol agents that I've mentioned. And you can find these publications and other publications on the eOrganic website or at the project website at swdmanagement.org. So why do we need biocontrol? So biocontrol actually can be a really useful solution to reducing the risk of insecticide resistance. On the screen are 2 additional publications that kind of go into detail about the risk of resistance for spotted wing. But if we include biocontrol agents in the management of our pests by introducing them into the natural areas that are adjacent to crop areas, those biocontrol agents in the natural areas will suppress the population of pests so that when the season for the cash crop comes in, there are less pests moving from the natural area into the crop area. And less pest pressure in the crop area means less sprays, and less sprays equals a reduced development of insecticide resistance. So these biocontrol agents can play a really important role in helping to manage resistance in spotted wing. So the type of biological control strategy that I'm talking about is called classical biocontrol. There's another type of biocontrol where it's augmentative. So every growing season, you have to reapply these biocontrol agents. But for us, classical biocontrol is the reconnecting of pests with natural enemies from the place where these pests originate. And that's what we're trying to do here with this OREI program. So researchers on this program went to the places where spotted wing originate from, and they found one parasitoid that does a really good job of finding and parasitizing spotted wing. The goal of our program is to deploy and release thousands upon thousands of these biocontrol parasitoids until they establish a population such that when spotted wing populations increase above the economic threshold, these populations will become established, and eventually, once they are established, their populations will begin to oscillate and sync with spotted wing populations. And that established population will not need to be augmented, but as it exists in the context of spotted wing management, the biocontrol will drive down the pressure of spotted wing below the economic threshold. And that's the goal of classical biocontrol. It's important to say that this type of biocontrol strategy takes years to actually establish these parasitoids in this new place, the US. So we so we're doing a really good job, and it's taking a really big effort to rear and release these insects, but we're going to need some time before we actually see them establish in the near future. But I have some data showing that it's actually starting to work that we're going to talk about in a minute. Here is a video kind of detailing the biology of Ganaspis. You see the Ganaspis finding the larvae inside of the fruit. Ganaspis lays its egg inside the larvae. The Ganaspis offspring then hatches and begins to develop inside of the spotted wing pupa. And once the Ganaspis becomes an adult, it will emerge from that spotted wing pupa to go off and mate and find its own larvae to parasitize. There's a video here on the right that shows, let me get it going. The video here on the right shows kind of a live action view of that Ganaspis parasitoid living inside of the pupae of a spotted wing. Since 2022, Ganaspis camara releases have occurred in all the states that are overlaid in pink. Those states are both ones that are a part of this program and ones that are releasing Ganaspis on behalf of the program. I want to take a second here and make an honorable mention for Leptopollina japonica. This is an additional biological control parasitoid that is also able to manage spotted wing. And lucky for researchers here in the US, there was an accidental adventive introduction of Leptopollina in British Columbia. And since that introduction, Leptopollina has been spreading across the US. These parasitoids are similar to Ganaspis in that they're able to manage Ganaspis populations. And lucky for researchers in the states where those Leptopollina have existed, they are going out and releasing Leptopollina alongside Ganaspis and monitoring the impact of the parasitoid that we brought back, Ganaspis, and this advanced population of Leptopollina as well. The OREI program is happening across 8 states with 9 teams that are a part of the program. That's the pink states here on this map. And those teams across those 8 states are looking to see if what we're doing is actually making a difference. And the way that they are doing that is by setting up paired organic berry sites, a control site and a release site. The release site is where the Ganaspis is deployed. Those sites are all identical in that they have a crop area, a non-crop area, where they collect samples, and the non-crop area is adjacent to a crop area, which transects at 1 meter, 10 meter, and 25 meters. At each one of these sites, whether it's control or release, researchers are collecting fruit, they're setting up sentry traps, and they're deploying sticky cards. The point of all this sampling is to determine the pressure of spotted wing in the area and to determine whether or not we're actually getting establishment of the parasitoid that we are releasing. So the next 9 slides all have the same format. It's a little bit about where the researchers on the team has set up their sites, pictures of the researchers, and a little bit of data, or some of the data that we've collected from each one of these teams. In Knox, Maine, those researchers have 2 release sites where they've deployed Ganaspis 3 times last year for a total of about 3,000 Ganaspis per site. They've been able to recover Ganaspis from both of their release sites. And in addition to Ganaspis, they're also recovering Leptopolina japonica. The bar graph below the table is the average percentage parasitism in the fruit collections that they're bringing back from these sites. And the important point to notice is that the proportion of parasitoids coming out of that fruit compared to Ganaspis, the large proportion is Leptopolina. So they're getting a lot of Leptopolina out of these samples. In Appling, Georgia, they had 1 release site. They released approximately 1,000 Ganaspis, and they were unable to recover any Ganaspis last year. The researchers there noted that the season last year was extremely hot, and they also recovered very little spotted wing. And without spotted wing, you can't have Ganaspis. So the heat may have played a role in their low recovery of Ganaspis. In Alachua County, Florida, they had 3 release sites. They made 2 releases of about 1,200 wasps total, and they recovered 4 Ganaspis. In Dakota County, Minnesota, they had 2 release sites. They made 5 releases at both of those sites of about 1,000 wasps, and they were able to recover Ganaspis at both release sites and one of their control sites. And just like Maine, the Dakota County, Minnesota, they were able to also recover a lot of Leptopolina from these sites, at all their sites. In Allegan, Michigan, the researchers had 1 release site. They released a total of 3,000 Ganaspis, and they're still processing their data to determine whether or not they've recovered Ganaspis from any of their samples. In Benton County, Washington, they had 1 release site. They made 2 releases of about 2,300 Ganaspis, and they have yet to recover any Ganaspis from those sites. In Lynn County, Oregon, they had 2 release sites. They made approximately 1,000, they made 3 releases of about 1,000 Ganaspis at each site. They've been able to recover 1 Ganaspis at one of their control sites, but just like Maine and Minnesota, they also were able to recover a significant amount of Leptopolina from their sampling. In Polk, Oregon, they had 1 release site. They released approximately 500 wasps, and they were able to recover about 58 Ganaspis from their release site, along with an additional parasitoid for spotted wing that's naturalized. Here in Monterey County, California, we had 2 release sites. We made 7 and 8 releases at each one of the sites. We released over 300 wasps at each release site, and we have yet to recover any Ganaspis, and we have not recovered any Leptopolina from any of the sites as well. So here's a table kind of summarizing what we found this year or last year so far. We have had GK recovery across 4 sites, or 4 states, excuse me, Maine, Florida, Minnesota, Oregon, and between control and release sites, Maine and Minnesota had 31% less spotted wing compared to their release sites, control sites. California had 28% more spotted wing at their release sites compared to their control sites. And for natural enemy populations, Florida had approximately 27% less natural enemies at their release sites compared to their control sites. And everywhere that data is available, bee populations between both control and release sites was similar. So in year 2, we were able to release a total of 29,456 Ganaspis chemorum biocontrol parasitoids. And we recovered those parasitoids at 10 sites so far. Across both of the years, we've released 34,494 Ganaspis and recovered from a total of 14 sites. One thing to note is that all of these sites where Ganaspis releases are happening are different. They have different climate, they have different environments. So that might explain some of the variation that we're seeing as far as recapturing some of these wasps in all of these different places. But that is all that I have. I'm going to now pass it over to Ben Johnson, who's going to talk about the compatibility of these parasitoids with OMRI-approved pesticides.
Hi, everyone. So as James said, I will be speaking on OMRI-approved pesticides compatibility with spotted wing and these parasitoids.
You should just be able to click on it once and yeah, okay.
There we go. Slight delay. Okay, so to start with, this is some of the research that came out of the first 2 OREI grants looking at impacts of OMRI-approved chemical control on spotted wing, looking at different phagostimulants that could be added to OMRI-listed insecticides, looking at adjuvants, looking at how long these pesticides work and last in looking for resistance, looking at how to measure for resistance in this pest. Just massive amounts of work to figure out what the best organic insecticides are to control this pest. And then these researchers all came together and ranked the insecticides for their efficacy against spotted wing. So these rankings range from no efficacy, which does not work at all, up to excellent. And it was made via a combination of lab bioassays, field trials, and some of both. And researchers came together and they discussed what they thought worked well, what does not work well. And you'll see the rankings. Entrust is the best option, and it barely reaches the good level of the OMRI listed materials. This was a collaboration from 9 states with 19 different state and crop combinations. And then researchers in these first 2 OREI grants also looked at the impact of these pesticides compatibility with natural enemies that were being found in fields where spotted wing were. They used 3 different natural enemies. This one shown here is a little parasitic wasp that's about the same size as the parasitoids we are releasing to control for spotted wing now. This one specifically attacks aphids. They also looked at 2 predatory insects, a little pirate bug and a lacewing. And they found that on the x-axis here, we have the different pesticides they were looking at, and on the y-axis, we have the percent mortality of these beneficial insects. So with this graph, the higher the bar, the more lethal these pesticides were for these good insects. So high bars are bad for the beneficial insects we see in these fields. Entrust, just as it was effective in killing spotted wing, was also effective in killing beneficial insects. But there are some of these other insecticides that were less lethal to beneficial insects. And the next work I'm going to talk about is a little bit of work I've done at the University of Maine, seeing how some organic insecticides impact Ganaspis and Leptoplena, these 2 new biocontrol agents that James was just speaking on. And I approached this from the viewpoint of these beneficial parasitoids living in the wild, not in fields, but in the field edges. And there are 3 main ways that I came up with that these beneficial insects could interact with spotted wing and insecticides in these untreated areas. So I've got a spotted wing right here. This cloud indicates a pesticide or some form of treatment, and then the beneficial insect. So in the first interaction, we have the spotted wing approaches the fruit, lays an egg and infests it, and then a treatment is applied to the field. And let's say there is some form of wind and a little bit of drift occurs and reaches the edge. And then you get the beneficial insect coming in and attacking spotted wing and parasitizing it. That is the first interaction. The second one could be a preventative insecticide being put on a field, and then spotted wing comes along and lays an egg, and then it gets parasitized. And the final one would be where infestation occurs, a insecticide gets, and then parasitism occurs, and then a treatment gets applied. So these three different interactions are what I was looking at. And I ran this in a lab. I had multiple different exposures. The main ones here that I'm talking about are the spotted wing plus treatment plus wasp, treatment plus spotted wing plus wasp, and spotted wing plus wasp plus treatment. In all of these bioassays, chemicals were given 4 hours to dry before adding insects. Spotted wing were left for 48 hours to lay eggs in fruit. Wasps were left for 48 hours to parasitize spotted wing. And then we added yellow sticky cards to count the emergence of spotted wing and parasitoids. These were run using a Potter spray tower, which simulates a spray in the field. And I tested this with 4 different chemicals, all organic, at a quarter rate to simulate drift or aged residues that a field might have. For the brevity of time today, I'm only going to be showing results from Entrust and peracetic acid or perioxacetic acid. These are your crop sterilants like Jet Ag, which work by influencing the microbiome on the fruit itself, as opposed to actually killing the insects. This is an example of what our little bioassays looked like. We had plastic deli cups with blueberries contained in a mesh cage. These berries were sprayed. We added the insects and then removed the insects, stored them in an environmental growth chamber set to 75 degrees Fahrenheit, 78% humidity, and a 16:8 light-dark cycle to mimic field conditions. So up first, we have our results from Entrust. There's 2 graphs on each of these slides. This first graph on the left, we have on the x-axis the different exposure order, and on the y-axis, we have deviation from control emergence, where the control would be a spotted wing infested a berry, and then a parasitoid attacked it and then emerged. So this is what you'd expect with 0 presence of insecticides. And the further away from 0 you get, the less parasitoid emergence we had. So with the spotted wing infesting fruit, and then a spray being applied, and then wasp parasitizing that spotted wing, we saw a large deviation from the control emergence. When a spray was applied, and then spotted wing infested the fruit, and then that spotted wing was parasitized by a wasp, we again saw fairly large deviation, but not as much as that first one. And then finally, when the spotted wing infested fruit was then parasitized and a spray was applied afterwards, we saw much closer emergence levels to that of a pesticide-free environment. On the right-hand side, we have the proportion mortality. So after adding the Ganaspis and Leptoplina, we looked at how long they lived in those bioassay containers for 48 hours. And you'll see with no insecticide, we were seeing 18% or 18% survival of the wasps. But once you start adding insecticides or chemicals to these environments, there is increased mortality of the parasitic wasps. You'll note this last one has a proportion mortality very similar to the control spotted wing wasp only. And that is because the treatment was applied after the wasp was removed from the bioassays. And this is the results from peracetic acid, your crop sterilants like Jet Ag. And you'll note that there was very little deviation from the control emergence. So peracetic acid had minimal impact on these beneficial insects. There is fairly large error bars, so we were seeing quite a bit of variation in this trial. And again, with proportion mortality, we were not seeing much mortality from peracetic acid in the adult wasps. And I should state that these graphs I'm showing are just for Ganaspis. We did run this trial with both Ganaspis and Leptoplina, as both are being found consistently in multiple states. And now I'm going to pass it off to Cesar to talk about non-target effects of insecticides with and without a phagostimulant on spotted-wing parasitoids.
Thank you, Ben. See if I got control of it there. So I will report on the work that we're doing to test the non-target effects of insecticides with and without a phagostimulant on the Spotted-wing Drosophila parasitoids. So although we are releasing the parasitoids in non-crop habitats, it is important to understand the non-target effects of commonly used insecticides against the Spotted-wing Drosophila on these parasitoids to better conserve them in our agroecosystems. So these are the collaborative states. These are the states where that have collected data on these non-target effects and send them to me. I know Betsy Beers has also collected some data from Washington State University, so I want to acknowledge that too. So as part of our efforts to develop behavior-based control strategies against the Spotted-wing Drosophila, we have done testing on a product called CombiProtect. This product contains sugars and proteins and acts as a feeding stimulant on a Spotted-wing Drosophila. So it can be combined with insecticide applications, and is currently registered for use in agricultural settings in some states in the US. So far, we only have 6 states where this is registered, but we're hoping that this will, the number of states registered will increase soon. So adding a feeding stimulant can increase the efficacy of insecticides by increasing the feeding rates of an insect. This can help reduce the amount of insecticides that are being used and also increase the residual activity of an insecticide. So CombiProtect can be applied in 2 different ways. It can be applied as a cover spray where CombiProtect is mixed with an insecticide and applied over the entire plant canopy or crop field. Or it can be applied as a bait spray where CombiProtect is mixed with an insecticide and applied in smaller quantities over reduced areas to maintain efficacy while minimizing the insecticide use. And previous studies have shown that CombiProtect can be applied either as a cover spray or a bait, can enhance the lethality of various insecticides, including entrust against Spotted-wing Drosophila. So this is data from New Jersey showing different rates of spinosad and with and without CombiProtect. And you can see that the addition of CombiProtect increases the efficacy of this insecticide. Similar studies were done in Georgia with similar results. So for the present studies, we asked, do different insecticide classes, rates, and residues have different impacts on Spotted-wing Drosophila parasitoids? We also asked whether insecticides containing CombiProtect induce higher mortality rates on these parasitoids, and whether cover sprays induce higher mortality rates on these parasitoids than bait sprays. So for this, we conducted, these are all laboratory studies where we tested 5 different insecticides. We looked at 8 different rates. So we had a full rate, a half rate, and a quarter rate, and a control. So no insecticide with and without CombiProtect. we had 3 residues per treatment. So we looked at 0 days after treatment, so soon after the insecticide had dried, then 3 days after treatment, and 7 days after treatment. For each test, we did 10 replicates, and we had 5 wasps per replicate. And we did this for males and females. So these are data that were collected from New Jersey. This, again, was repeated in other states. This is data that was collected by my master's student, Jack Collins. These and all figures in the following slides present the percent mortality on the y-axis of Aganaspis skimmorum exposed to an insecticide at the full, half, and a quarter rate, and compared to the controls in blue. The data for females are shown with the solid lines, while the data on males are shown in the dashed lines. So this is the data just with the Entrust alone. You can see that Entrust had some mortality on Aganaspis skimmorum. And when we mixed it with CombiProtect, we observed some reduced mortality. This is the data for the 3 days after treatment. So again, Entrust continued to have some mortality on Aganaspis skimmorum. However, when we mixed it with CombiProtect, we actually saw an increase in mortality. This is the data for Entrust at 7 days. So we had lower mortality than at 3 days after treatment. And when we mixed it with CombiProtect, we saw again an increase in mortality. This is the data for Exirel. For Exirel, we had less mortality overall compared to Entrust. And we observed little effects when mixed with CombiProtect at 0 DAT. Again, at 3 days after treatment, Exirel had fairly low mortality, but when mixed with CombiProtect, we saw an increase in mortality. This is for 7 days after treatment with Exirel alone, and with Exirel with CombiProtect, we saw little effect. So Mustang Max, this was the more lethal of the 3 insecticides that I'm showing you. You can see like mortality was fairly high on Aganaspis skimmorum with and without CombiProtect. After 3 days after treatment, the lethality was lower. But interestingly, the mortality decreased with the CombiProtect. And we observed the same thing at 7 days after treatment. When we mixed with CombiProtect, we saw a decrease in overall mortality. So we, as I mentioned, there's 2 ways to use this product, this CombiProtect, either as a cover spray or a baked spray. So we wanted to see whether the baked spray had less lethality on the insect. So we did a study, similar study with 5 insecticides. We looked at different rates with and without CombiProtect. We looked at 3 residues per treatment, 0, 3, and 7 days after treatment, 10 replicates, 5 wasps per replicate, and also we looked at males and females. So this is the data for Entrust without the CombiProtect. It was fairly similar results with and without the CombiProtect. This is for the 3 days after treatment with Entrust alone. And with the CombiProtect, we saw an increase in lethality, as very similar to the cover spray. And this is for 7 days after treatment with Entrust alone. And again, when we mixed it with the Combi Protect, we saw an increase in lethality. Very similar results to the cover spray. Xeral, again, was the least lethal of the 3 insecticides that I'm showing you. With 0 days after treatment, there was not much of an effect with Combi Protect. This is 3 days after treatment. We saw an increase in the lethality when we mixed with the Combi Protect. And with 7 days after treatment, we saw little effect. This is for Mustang Max. You could see that the lethality of Mustang Max was reduced when used as a bait spray. There was little effect on the Combi Protect at 0 days after treatment. With 3 days after treatment, we saw a decrease in lethality with the Mustang Max. And we observed the same at 7 days after treatment. So, what are we finding? So to answer our question, do different insecticide classes, rates, and residues have different impacts on Ganaspis skimorum? We are seeing that yes, different insecticides had varying lethality effects. So Mustang Max was the most toxic, followed by Entrust, and Xeral was the least toxic. Also, we saw that lower insecticide rates induced lower mortality. So the full rate was, as we expected, the most lethal of all of them, higher than the half, and that was higher than the quarter. And the treatments that were aged for longer induced less mortality. So 7 days after treatment had the least lethality that compared to 0 days after treatment. So to answer our second question, do insecticides containing Combi Protect induce higher mortality rates on Ganaspis skimorum? So in general, what we observed was that Combi Protect increased mortality when mixed with Entrust and Xeral, especially like we observed this for males. Males were more susceptible overall to the insecticides than females. In contrast, Combi Protect tended to decrease the lethality of Mustang Max. However, as I showed, Mustang Max was the most lethal of the 3. And this could be due to the different modes of action. Mustang Max is absorbed through contact, not by ingestion, whereas Entrust and Xeral have to be ingested. And similar effects of Combi Protect has been seen for Spotted-wing Drosophila. This is a paper that we submitted for publication. So to answer our third question, do cover sprays induce higher mortality rates on Ganaspis skimorum than bait sprays? In general, the answer is yes, but mainly for females that were exposed to Entrust and overall for Mustang Max. So our future directions is to evaluate these insecticides and these non-target effects under more realistic field conditions. So with that, I want to thank the participation of the Philip Alampi Beneficial Insect Rearing Lab in New Jersey, who provided us with the Ganaspis skimorum for these bioassays. And with this, I pass to Philip.
Okay, thank you, Cesar. Okay, so I'm going to round out this presentation before the questions with talking about determining the compatibility of organic compounds, more specifically determining the compatibility of nematodes with Ganaspis skimorum. We have looked at the impacts of, or there's been many research trials looking at the impact of entomophilic nematodes on Spotted-wing Drosophila. As you can see here in this table, there's been multiple different species that have been looked at. You can see all of the different trials and whether they were successful or not. So 2 that we used in this experiment was Steinonema carcapsia That has been successful in 20 out of 23 trials. And then Steinernema feltii, that has been successful in 18 out of 21 trials. I should back up and say that I must thank my colleague, Dr. Jana Lee from USDA for developing these slides, and I'll present them today. Okay, so when we think about where nematodes might fit in on controlling Spotted-wing Drosophila, we can think about basically the life cycle of Spotted-wing Drosophila. Obviously, you have your adult flies. They lay their eggs into the fruit. They develop through their larval instars. Sometimes there is pupation in the fruit, and then new Spotted-wing Drosophila will emerge from those puparium, and the life cycle begins again. But many of those larvae will drop out of the fruit into the soil where they'll pupate after being basically what we'd call a wandering larvae, where they wander around for a bit. So the goal of nematodes is to apply it to the soil to target those wandering SWD larvae and the pupae that are formed from them. So what happens when nematodes are applied to soil and encounter parasitoids, larvae, and pupae? This is what our experiments that we were hoping that we were running here were to determine. So we have our life cycle of Spotted-wing Drosophila. We have our parasitoids parasitizing into those larvae and the pupae. And we are basically challenging them with nematodes to see how that impacts the survival of both Spotted-wing Drosophila and our Drosophila parasitoids. So when we think about this, this was a team effort. It was conducted across multiple states. You had the University of Maine, University of Georgia, University of Florida, University of California, Davis, and USDA IRS in Carvallis. We had multiple researchers working on this, including multiple graduate students. When we think about our methods, so we were basically infesting fruit with Spotted-wing Drosophila. So this was done in the laboratory under kind of laboratory conditions. We would infest fruit. We would introduce those infested fruit into arenas, and we had 4 treatments in total. The first treatment was wasps and entomophilic nematodes. The second treatment was wasps only. The third treatment was EPNs or entomophilic nematodes only. And then we had a control where we just had Spotted-wing Drosophila. So we would infest that fruit. We would count the number of eggs in that fruit, which can take quite a while and can be very laborious. And then we would put them randomly into their different treatments. In the treatments where wasps were being included, we would include wasps, so treatments 1 and 2, and where no wasps were included, that fruit was just left at the same conditions without wasps being included. After 7 days, that fruit was basically transferred into a petri dish with moist sand, and then we either added the entomophilic nematodes or the water to those wandering larvae or those early pupae. They were then held until about 15 days, where we started counting basically how many Spotted-wing Drosophila emerged. We dissected the puparium or the flies, and we also counted the number of wasps that were emerging from treatments that had wasps. In total, we used 1,800 infective juveniles per arena, which was deemed to be an effective concentration in previous studies. Now, we didn't do this just on larval parasitoids. There was research done on pupal parasitoids, specifically Pachyploides vindimera and Trichopia drosophila. These are common parasitoids in the United States, and they have been found to parasitoid Spotted-wing Drosophila puparium. We did a similar methodology from them, where SWD was introduced into an artificial diet. Those SWD laid eggs. We allowed them to develop as far as puparium. And we did a very similar trial to the one I just described, where those puparium went through those four different treatments of either wasps plus EPNs, wasps only, EPNs only, or an SWD control. Similarly, we use the same rates of spotted-wing or of the EPNs, and we had the same count of Spotted-wing Drosophila, parasitoids, and dissections. So this was mostly done on two species of EPNs, Steinonema carcapsia. Some notes on Steinonema carcapsia are seen here in blue. So these can also infect new SWD larvae as they emerge from pupae. They're generally found in the top 1 to 2 centimeters where SWD pupae are often found. The other species tested, which was Steinonema feltii, this can kill SWD faster than any other nematode. So these are two important nematode species which we thought would be good to include in this research. So the first question, can nematodes work with parasitoids? And the answer is yes. The combination of wasps and nematode kills more SWD than either alone, at least in 3 cases, leading to about a 56 to 83% reduction relative to that Spotted-wing Drosophila only control. Or the combination treatment had equivalent SWD emergence as the wasp only treatment. So you can see all of the different treatments we had here in this graph in the bottom. On the y-axis, you have SWD emergence, so from 0 to 100%. And then on the x-axis, we have all of the different parasitoids and EPN combinations. So we looked at Ganaspis camorin, Leptoplena japonica, Pachyploides vindemia, Trichospia drosophilina. And they were all tested with Steinonema carcapsia. And then Ganaspis camorum and Leptoplena japonica were also tested with Steinonema feltii. In general, in this graph, and I won't walk through it too much, the lower bars is better. So the lower emergence is what we want to see. Now, of course, the next important question to ask is, does nematode exposure lower wasp survival? And unfortunately, the answer is yes. So you can see here in the wasp versus EPN treatments, which are in the blue bars, and then the wasp-only treatments, which are in the orange and white bars. And generally, we had lower wasps emerged in the combination treatments. So what would that mean? So how would we basically navigate or use this as a recommendation? If parasitoids are already there, nematodes might help further suppress Spotted-wing Drosophila. Jana and others are currently running field trials looking at the delivery of nematodes via drip to continue some of this field relevant research. Now, if your goal was to release parasitoids and get establishment, then maybe avoiding applying nematodes in that area would help increase establishment. Okay, so in general, over take-home messages from this research, Spotted-wing Drosophila, still a challenging pest in organic fruit production. There are limited pesticide options, but we do now have classical biological control options, and we have these other biological control options, which we're working on integrating together. These classical biological control options will be important in reducing the impact of SWD on organic growers' production. However, we are learning that pesticides and nematodes can have negative impacts on Drosophila parasitoids. So our goal and our future directions will be to continue this research to try and best establish these parasitoids, suppress SWD, and integrate all of these biocontrols into consistent programs for the control of Spotted-wing Drosophila. So one thing I also wanted to mention as part of this experiment, we were also running a research and extension experience for undergraduates. A lot of these projects require a lot of research, and generally undergraduates really help us complete this research really successfully. part of this project, we also had built in research extension experience for these undergraduates, where they would go through training throughout the summer as they worked in our labs. This was led by Dr. Mary Rogers in the University of Minnesota. You can see here, this is our class of 2023. Here's our class of 2024. These researchers or undergraduate researchers put in a lot of effort. They presented their research at the end of the year. There's some of the information on their research projects on eOrganic, but it's important to note that this is really a big group project, and it's got input from a lot of really amazing young researchers that we're hoping will develop and continue. So with that, we'll acknowledge our grower cooperators. I know James presented a lot of information from different farms across the country, and a lot of those grower cooperators were very great and willing to work with us. We'd like to thank all of our state and crop commodity groups who've helped us give input. And then also, finally, we thank our funding sources, which is USDA and IFA, and the Organic Agriculture Research and Extension Initiative. And with that, we will take any questions you might have.
Okay, thank you very much. Yeah, this relates to the beginning of the webinar. Did you have any results on the crop losses for grapes? How are wine grapes affected by spotted-wing Drosophila?
Yeah, when initially that survey was ran, wine grapes were not considered. It was very much thought that grapes generally weren't as impacted by spotted-wing Drosophila as some of these other small fruits. We do know spotted-wing Drosophila can impact grapes, especially where that skin is already compromised. Spotted-wing Drosophila can then oviposit into it. But we were only really considering those other fruits in that original survey, and then we're following it up just with those ones that we originally surveyed.
Okay, here's a question about whether there's somewhere right now that growers can buy Ganaspis camorum or the other biocontrol agents you were discussing.
Currently, no. There are some labs thinking about developing, we'll say, rearing for it, but currently most of it is done by our labs purely for research purposes, so they're not commercially available currently.
Okay, let's see. These are two questions for James. I'll ask them one at a time. How many days apart are Ganaspis camora releases in the treatment and the control areas?
That schedule is determined by the team that's working on it. I know for us, when we do releases, we stop trapping for spotted wing and then we resume after a couple of weeks. But we had a schedule of releasing. The goal was to release every time we visited our sites on a weekly basis, but that tends to fluctuate with the population that's available that we're rearing. But I would say probably somewhere between 1 week and a month, we put out wasps into these areas.
Okay. Have you noticed any kind of synergy between the parasitoids you release and those naturally present in the environment near the orchards?
So I'm gonna have to pass this question to someone else. Unfortunately, in California, we have not recaptured Ganaspis last year. And while I was in Florida, we only recaptured Ganaspis a few times. And we weren't recapturing a lot of natural enemies that could synergize with these parasitoids. I know in other places like Maine with Phil's group, they are seeing a combination of Leptopolina and Ganaspis in their traps, but I should probably let him describe those situations.
Yeah, I think the best kind of data we could point to on this is from when the original, we'll say, research was being done to find these parasitoids in the explorations that were done in Asia. In general, these two parasitoids were found in the same samples. So it's generally thought that they do play well together, if we could say that. Now that might change if we had we'll say a situation where there's a limited amount of hosts. But in the original survey work that was done, these two parasitoids were generally found in the same fruit, infested fruit. So we do think that they are able to play well with each other.
Okay. Is there any augmentative biocontrol worth doing in the field that can be done with currently available natural enemies while we gather more data and establish our Ganaspis and Leptophilina populations?
Yeah, I think that comes back again to commercial availability. Unfortunately, I don't think even some of those parasitoids, some of the pupal parasitoids, are available commercially just yet. I think in terms of commercialization of parasitoids, um, a lot of effort goes into rearing these in all of the labs that are taking part in this. Rearing them is really quite difficult currently, and it's quite expensive. Um, Leptopolina japonica is probably the easiest of all of these parasitoids to rear because we can rear them on commercial diet, etc. But it can, currently we don't have release permits for Leptopolina japonica, so they could be distributed nationally.
Hmm. Okay.
I know that,
Okay, sorry.
sorry, I know that Trichopria Drosophila, like they sell them in Mexico and parts of Europe, but not in the US. But data have shown that these people parasitoids are not as good as the larval parasitoids. So that's why we are like working mainly on Ganaspis and Leptolina.
Okay. Let's see. Here's a question about nematodes. If nematodes live in the soil, why would they impact parasitic wasps? Don't wasps pupate inside their host?
Yeah, so it's capturing that wandering phase. So once the larvae drop out of the fruit, before they pupate, they generally will interact with the soil. So if nematodes are present, we can target them at that juncture where they're wandering around the soil. Um, Even in puparia, they could possibly enter puparia, especially if that puparia had been compromised, maybe by a predator, etc. So there could always be possibilities for, we'll say, those entomopathic nematodes to get into the puparia as well.
Okay, going back to Heather, who asked a question about the augmentative biocontrol. She was talking about Aureus, Delocia, etc. Would it be worth doing those in the field before the other ones that you introduced are established?
Yeah, that's a really good question. I'm not sure of any experiments or trials that looked at augmentative aureus releases to control Spotted-wing Drosophila. But we could certainly look them up and get back to them if they reach out to me via email.
Okay, great. Let's see. If is there any do you have any speculation if pesticide applications can affect nematode efficacy for spotted wing Drosophila biocontrol?
Yeah, I think there was one question, or there was an answer to that by Dr. Jana Lee
Okay.
in the chat. So some
Oh,
pesticides
okay.
can affect nematodes in lab studies. In the field, it may depend on how the insecticide is applied, how much it reaches the soil, where the nematodes most likely are applied.
Okay, great. Thank you, Jana. Okay, let's see. I've got to go back here in the chat. Okay, yeah, this is a question about commercial availability. I'm gonna go down here. Let's see. Okay, can somebody briefly address the seasonal phenology of spotted wing Drosophila regarding timing of nematode applications?
Yeah, so I think generally when these nematodes would be applied, you'd want to be applying them as spotted-wing Drosophila populations are starting to build up. Obviously, for them to be effective in the scenario and the life cycle I showed, there would already be some spotted-wing Drosophila in and around the crop. Pupae would be present, and we'd ideally be trying to stop those pupae that are in the crop from, developing to adults. So it would probably happen as your SWD population is building up. So that would require monitoring your SWD adults in and around your crop at the same time, which we recommend, of course, obviously.
Okay, we have a question here from a grower. Could you use additional demonstration sites for releases? We have a small farm surrounded by native vegetation or getting a complete loss of our blackberries. We'd love to help with this project, and I think we have a good potential site. Are you looking for more grower collaborator?
I guess it would depend what state they were in and how close they are to our, we'll say, the nearest tier member. If they reach out to me, I can direct them to who to talk to.
Okay, great. Okay, we have a comment from Kent Dane. Work in Mexico, California, and Italy suggests that there may be some potential for releasing the pupal parasitoids, Trichopria Drosophila, and there are, let's see, oh, I don't know, the question just skipped around here, so I got to put it in full screen, so I've got to go back to the comment. Let's see. Oh, yeah. Okay. Trichopria Drosophila, and there's some rearing operations such as Driscoll's who are testing this out. Thank you, Kent. Okay. And okay. So Heather says maybe she has a research project in her future about these biocontrol agents. So thank you. Okay, so do spotted-wing Drosophila overwinter in agricultural fields? In other words, would it be beneficial to apply nematodes in the off-season to decrease overwintering populations?
I can address that a little bit. I guess that depends greatly on where you are in the United States. We, on other projects, have done a lot of work on spotted-wing Drosophila overwintering. In general, we think they overwinter as adults, and we do think they overwinter in the leaf litter down in the soil. Whether it would be beneficial to use entomophilic nematodes in the off season, that would be a good question. A lot of these EPNs are also impacted by temperature, so their activity is likely lower when temperatures are cooler. So targeting late season spotted-wing Drosophila as they overwinter, you might not have as good an efficacy purely based on the fact that, colder temperatures at those points would likely impact the EPN's ability to search out spotted-wing Drosophila overwintering. But it could be a potential.
Okay, great. We have a comment from Luis from Chile, who said that they are releasing Trichopria Drosophila in the field weekly from January to March 2025.
Excellent.
Thank you for that. Okay. Well, let's see. That looks like all the questions in the queue. So if you have any more questions, okay, good. We have another comment from Jana here that Aureus and Delosia have been tested in smaller arenas. We did not test Aureus in an open field trial. Justin Renkema checked Delosia for predation on spotted-wing Drosophila by molecular tests. So thanks for that comment, Jana, too. If anyone else has any questions or so, let's just leave another minute in case anybody else would like to ask a question of this group or make any more comments. I just wanted to remind you all again that we recorded this and you'll be able to find this and more information about spotted-wing Drosophila and organic strategies to manage it on our project website at eorganic.info/spotted-wing-organic. And also on the eOrganic YouTube channel, you can find many other recorded webinars on spotted-wing Drosophila organic management strategies from this group from past NIFA OREI projects. Okay, so last question here. Do you have an estimate for how long until the commercial availability of these parasitoids in the United States?
Yeah, it's a difficult question to answer. I think we're still working on figuring out some of the rearing methods. And once rearing methods are, a lot easier, maybe it might be more commercially viable for, we'll say, some of the companies that provide natural enemies to jump on board and start doing commercial kind of releases or offering it as a product. But I'm not sure on what the timeline might be or what, we'll say, some of those companies' appetite might be for developing this as a commercial product.
Okay, well, thank you very much. It looks like we have answered all the questions. So I'd like to thank you so much, Philip, Cesar, James, and Ben, for sharing your research with us today. And we look forward to more webinars from this group. So thank you all for your questions, and thanks everyone for joining us today.